Hi everybody, so my name is Fran Gallardo. I'm the CEO of a company called NeoVirTech. We are localized in Toulouse, France since 2014, and our main activity is to look for compounds that will impact infection in cells, and we use a unique technology that we have developed back in 2010 that is called the NeoVirTech technology. We work on several kinds of viruses, viruses that can impact the human health, the biodefense market, but also the veterinary market. We have several confinement laboratories, so for example we have BSL-2, BSL-2+, and for highly pathogenous viruses such as influenza viruses and SARS-CoV-2, we have a BSL-3 lab. The technology that we have developed is called the NeoVirTech technology. Is based on two parts. The first part is called the ENG sequence. It's a sequence that you insert inside the virus that you want to image. And then the other part is the R protein. It's a protein that is fused to a fluorescent protein that will accumulate into the ENG sequence and will allow you to visualize the infection and the replication of the virus in living cells. So this technology has been applied to different kinds of viruses. For example, we have herpes viruses, adenoviruses, we have a large collection of pox viruses, and recently we have been the first to be able to visualize retrotranscription and integration of HIV inside living cells. So our asset is that we use the NeoVirTech technology, which is an image-based technology. It's not a classical virology technique. We generate a lot of information using imaging, so we have to use machine that allow us to image a lot of conditions in parallel, and we use high content screening microscopes such as the CX7 from Thermo. So this microscope allows us to compare a large number of conditions simultaneously and to identify new molecules that will have an impact in the infection, and this molecule can be next generation antivirals. When you use the NeoVirTech viruses, you can score two parameters. You have first the infection rate, which corresponds to the number of fluorescent cells per well, and in each infected cells you have the replication rate, which corresponds to the accumulation of the NeoVirTech signal inside the viral replication center. So we use a spot detector algorithm that allow us to compare directly the efficiency of a molecule of interest. What we saw for the COVID-19 crisis is that it's not only the research on SARS-CoV-2 and coronaviruses that were boosted. All the virology came in light, and we have a lot of requests to test for other methods to disinfect, for example gloves, surgical masks, etc., etc. But also the problematic of viral persistence inside the environment, and to look for innovative solution in disinfection has come in light. So this is now a major part of our activity at NeoVirTech. So for the future, which is almost the present, we have developed two new methods. The first one is that we have been the first to develop the high content screening methods for SARS-CoV-2 infection in human pulmonary cells. So we are able to investigate the impact of a compound on the infection rate of SARS-CoV-2, but also on the viral RNA replication level, and we do that in high content format. The second thing is that we have developed a technology that allows us to contaminate living human skin explant, and then to look at the impact of a solution, disinfection solution such as hydroalcoholic gel solution or disinfection wipes, or also intelligent surfaces. And this allows us to determine the disinfection potential of this solution on the viral load, for example, so we can calculate a decrease in the viral load but also in the impact of this product on the skin physiology, and this is done with a company called Sentivia here in Toulouse. We also change our view of the virus infection. Everything that we've done so far was done in 2 dimensions. Now we are going in 3-dimension infection using spheroids, and this is done using the CX7 in a confocal mode. But we have also access to a light sheet microscopy with our colleagues from Imactive 3D, which allow us to go really deeply inside the infected spheroids and to understand the behavior of the infection in the 3-dimension environment.
Trop, ça.
Et alors